A very good afternoon to everyone gathered here. As we are waiting for more participants to join the webinar, I would like to give a brief introduction to the webinar series that we have planned in this program. So as part of the Silver Jubilee celebrations of Shastra deemed university, the Division of Bioinformatics and the DBT-sponsored Bioinformatics Center and the Computational Biology Group at Shastra, the Students Association called as COMBIX, are all organizing a webinar series on the theme Connecting Experts and Advancing Knowledge in Bioinformatics and Computational Biology. So this series aims to bring together distinguished experts from both academia and industry to discuss key advancements in the frontier areas of bioinformatics and computational biology. With this brief introduction, I would like to invite the Dean of the School of Chemical and Biotechnology to offer his welcome remarks.
Yeah, thank you very much, Professor Ashok, for the short introduction. It's my pleasure to welcome everyone for this webinar series, which has been organized by the School of Chemical and Biotechnology, in which the bioinformatics is a part of. And as Professor Ashok pointed out, it is an event which is co-organized by the three different entities: the Department of Bioinformatics, then the Bioinformatics Center, which is supported by DBT, and also the Students Professional Association, which is the COMBIX. I would also like to place on record the support of the government to the Bioinformatics division at Shastra University. So the Department of Bioinformatics at Shastra University had its beginning in the academic year 2002-2003, which means this department has now completed 20 plus years of its existence. So the first program that the department offered was an MTech program in bioinformatics, which was started in the year 2002 with the support of the Department of Department of Biotechnology, sorry. Subsequent to DBT's support and also the successful conduct of undergraduate and postgraduate programs at our department, the department was also recognized by the DST under the FISH program. And subsequent to that, we also successfully established a bioinformatics center. So this is a journey that the bioinformatics division at Shastra University has taken, starting with the support of DBT, then getting supports from the corporates for under the consultancy grant, then DST FISH support, again coming back to DBT to establish the Center for Bioinformatics Center and the National Network Project. So during this 21 plus years of our academic journey, okay, we have learned with a lot of students, we have learned with fellow experts, and it is also time that we give something back to the community of bioinformatics. And as a result of which, in the last 3-4 years, we have been consciously organizing several scientific events to ensure that the School of Chemical and Biotechnology and the Bioinformatics Division acts as an epicenter for knowledge sharing in the field of bioinformatics. And this webinar series is also going to be one among those such efforts, and it also one of the programs that we are organizing as the part of 40 years celebration of our institution. So without taking much time, I would like to hand over the stage to the first speaker for this session. And Professor Karthik Raman has been a very good supporter of our institution, and I am also very thankful for accommodating. I'm very thankful to Professor Karthik Raman for accommodating many of our students as interns in his labs. So we look forward to your talk, sir. Thank you very much for accepting our invitation, and the stage is now for you.
Thank you. I hope I'm audible and you're able to see my screen. So thank you very much. Thank you very much.
I'm just sorry to interrupt. Shall we have a speaker introduction? We have a student who will introduce you, so that the wider audience, I think everybody knows you, but still, I think it's in order. So shall we just call?
Sure, please.
So Ms. Subiksha from third year Bioinformatics, could you please introduce the distinguished inaugural webinar speaker for today? Thank you.
Subiksha, you're mute.
We can't hear you, Subiksha.
Yes, sir. Can you hear, sir?
Yes.
Yeah, fine now. Yes.
It is my great honor and privilege to extend a warm welcome to all of you to joining for us today's webinar, proudly presented as Combics series. We are delighted to have Professor Karthik Raman from IIT Madras as a distinguished speaker. He is a professor at Vadwani School of Data Science and AI, co-coordinator for Center for Integrative Biology and Systems Medicine, and also a core faculty member at the Robert Bosch Center for Data Science and AI. Professor Karthik's research group, Raman's lab, works on developing scalable algorithms and computational tools to understand, predict, and manipulate complex biological networks. Key research areas in his group encompass microbiome systems in biology, in silico metabolic engineering, biological network design, and biological big data analysis. Professor has also authored a textbook on computational systems biology. Professor has been recognized with several awards, to name a few. Institute Research and Development Award Early Career IIT Madras for outstanding achievements in teaching, scholarship, and creative research work. Young Faculty Recognition Award for excellence in teaching and research. Professor Karthik has an impressive academic portfolio with over 70 international publications to his credit. For today's webinar, Professor will deliver a talk titled Algorithmic Adventures in Microbial Ecosystem, Disentangling Complexities with Metabolic Modeling. So, without further ado, let us begin this webinar. Please welcoming in Mr. Professor Karthik Raman. Thanks.
Thank you. Thank you for the kind introduction and thank you for this opportunity. I'm very happy to be at Shastra virtually. And I do have a close association with Shastra and not only interns, as the Dean was remarking, several of my PhD students are from Shastra, so I'm quite indebted to Shastra for this, for bio research program, so to speak. So with that, let me just quickly go into this very interestingly titled talk, I hope, like Algorithmic Adventures in Microbial Ecosystems. And a couple of high-level agenda items for me is that I want people to start thinking about computation, mathematics, and biology in a single mind, right? So for too long, I think we've been trying to diverse the two because I either like mathematics or I like biology, but I keep insisting that people should like mathematics and biology if you need to work in this kind of an exciting space of computational biology. So I think a lot of has already been said in the introduction, but let me just quickly tell you a little bit about ourselves so you know where I'm coming from. We are a very interdisciplinary group focused on the development of computational tools and scalable algorithms. These are some of the interesting problems that we worked on in the past, from microbiome systems biology to designing regulatory networks to in silico metabolic engineering, biological big data analysis, and so on. And driven by a lot of methodology, including network biology, constraint-based modeling. These two I'm going to go and talk about it in a little bit of detail today, but also machine learning and high-performance computing. And we like to understand and manipulate biological networks. We work with experimental collaborators who try to do the metabolic engineering strategies that we predict. They try to implement it in the wet lab and so on. And we also perform integrated analysis of multi-omic biological data in the context of cancer genomes and so on, with the ultimate goal of impact in the clinic, industry, or the environment. And we actively drive the Center for Integrative Biology and Systems Medicine, which is also a very core part of the Vadwani School of Data Science and AI. So there are lots of algorithms and applications that we've developed over the years, and I'm going to touch upon a few today. But I'm mentioning the sort of wider portfolio of our research here so that it may interest some of you to go and look it up. Particularly, I would like to draw your attention to Metas at Chennai. We should get a preprint out in like a week's time on this. So we've sampled the Chennai Metro Rail for its microbial signature and find how it's different from what has been reported in other cities across the world, and things like that. So with that, I think almost nobody requires an introduction to the microbiome today, but still I think there is, I presume there's a very wide audience today with a varied background, and I think we must get over this notion that microbes are like bad or enemies and things like that. I just walked out of a memorial lecture today on phages. And the speaker there was saying something very similar in terms of, we always think viruses as being the bad guys. You probably remember, you recall the COVID virus or the polio virus and so on. But there are so many benign viruses and helpful viruses and so on. So similarly, microbes are all around us, and well, of course, pathogens take up a lot of our mind space and concern and so on. They just are very important for so many other activities and huge impact on human life, sustainability, and the environment. So microbes thrive in multi-species dynamic environments where the community structure is dictated by a very complex web of interactions. And this is what we like to draw on, that the metabolic interactions typically predominate here. So it's very important to understand what kind of metabolites these microbes produce and how it impacts their interaction with each other and with the environment and so on. And microbial consortia and co-cultures are useful in many applications like in metabolic engineering and so on. And understanding these interactions is crucial, whether it be to develop a new microbial co-culture for producing, say, a vitamin C or something like that, or if you want to design a new probiotic to recover the gut of a person who has inflammatory bowel disease or something like that. So in all of these cases, there are microbes, there are microbial interactions, and there are metabolic interactions between their microbes. And the idea is, can we study these interactions and ultimately understand and manipulate them? So how can we understand microbial interactions in communities? The question is, can we use metabolic models? So metabolic models have been around for more than 2 decades now, and typically started off with modeling single microbes, single bacterial cells, and so on. And now we've been building models to extend these to communities and how you can use these to understand community structure and function. So you might have seen these kinds of images in either a textbook or on the wall of a biochemistry professor or elsewhere. Right? So this complex picture that you see on the right is like what I typically call a simplified representation of the complex metabolic network that underlies every living cell. Right? So this is simplified because simply there are several, say Avogadro number of molecules, or like say 10 to the 15 molecules of each species, and they all collide and interact in so many different ways in this very complex web of metabolism within the cell. And these present a comprehensive, so these models try to capture a comprehensive picture of all the known metabolic reactions or transports happening within a cell. And we can build these things from genome sequences today. So you can build what are known as these draft reconstructions from genome sequences, wherein you can punch in a genome sequence into one of these websites like KBase or ModelSeq and so on, ModelSeq, and they will help you generate all the list of reactions that are possibly happening in that organism and things like that. And using that, you can use an array of computational techniques to simulate these systems. Ultimately, this is what we like to think of it, right? So reduce it to some sort of a network with like nodes, edges, and so on, and try to understand the system from this perspective and use it to build models. How do we model microbial communities? And there you go, you have a Shastra alumnus here, Dr. Aarti from my lab. She was my first PhD student. And together we wrote this nice monologue on how do you, model microbial interactions using different kinds of methodologies, right? So there are constraint-based models, there are graph-based models, which I will talk about today, and also population agent-based models and so on. And there are many more methods being developed, but all along I would like to emphasize that the metabolic interactions and exchanges are of particular interest, and modeling them helps us get a good handle on what's actually happening between these microbes in reality. So how do we model? So constraint-based modeling is a very popular method. I have long ago probably lectured about it in Shastra. It's very popular for applications in metabolic engineering. But just think of it as a simple set of techniques that are rooted in linear programming and having linear constraints on mass balance and things like that to predict what is possible and what is not possible within a cell. So just as a at a high level. In network-based modeling, this is graph theoretic modeling and so on. So all of you are familiar with networks and you've been using networks even if you haven't been realizing them. It's like it's like the Google Maps within a cell. So in Google Maps, you're always trying to find the shortest or fastest path between two nodes in a graph. So graphs are networks, so where nodes are could be places or junctions and so on, and edges are various kinds of roads and things like that. So this is what you have in Google Maps. And the same thing is extended to a metabolic network where you have the nodes as metabolites and the reactions as edges and so on. There are a few different ways to represent them, which I will talk to you about in a moment. So this is a broad overview of some of the algorithms that we've developed. So we'll start with identifying microbial interactions in communities, where we have an algorithm called MetQuest. This small icon on the side is just to emphasize that this is a network-based approach as opposed to constraint-based modeling approaches, which I will also be presenting today. And you will see that all of our lab work is open source, and you can freely download our algorithms and software tools from GitHub. And we have other people contributing to them as well. You're also welcome to look at it, tinker, and so on. So how do you convert a metabolic network to a graph? As I already gave you the Google Maps example, you have this notion of nodes and edges, right? So what should be a node? What should be an edge? So I have grayed out a few which are not so useful, but here you may be able to observe that there is glucose which is getting transformed to glucose 6-phosphate and so on. But in this kind of a representation, there is a challenge. You may be able to convert glucose. You may think that you're converting glucose to ADP and ADP to pyruvate, resulting in a two-step glycolysis, which is absurd. So what we need to have is a different kind of representation, which lets you know that you need more metabolites to produce a particular set of more substrates to produce a particular set of products. So here you have ATP and glucose that, let's say, go into hexokinase, and then out come glucose 6-phosphate and ADP. So this is what is known as a bipartite representation. So you have two types of nodes. So you have circles and squares, and there are no connections directly between circles or directly between squares. And it's all between circles. Circles connect to reaction squares, and reaction squares connect to product squares, and substrate squares connect to reaction squares, and so on. Substrate circles connect to reaction squares, and so on. So this is a very nice way to represent metabolic networks. So this is called a bipartite graph, and we use this representation to encode metabolic networks for pathfinding and so on. So what does our algorithm use? It uses this bipartite representation. So we have the same thing, no? We have circles and squares here, and we use what is known as a dynamic programming approach, which is a sort of common approach in computer science. But we tweaked it to our scenario because you have certain unique aspects in biological networks and so on, to ultimately assemble pathways of a particular size from a given set of source molecules. So I have like some food molecules, let's say like there is some glucose and so on. And then from this glucose and maybe some amino acids and things like that, how is the organism able to produce molecules required for its survival? So we map out these pathways. And how do you model communities? You have this notion of two organisms or multiple organisms. And here you have C1, A1, B1, and so on. So the metabolites of organism 1. And here you have C1, C2, A2, B2, and so on. These are the metabolites of the second organism. And these are exchanged through a common pool, like an extracellular space and so on. So B1 is secreted out to B, and then B is then taken up by organism 2. So B1 ultimately comes in as B2. And maybe the presence of B1 actually enables A to do some more metabolism and things like that. So these are the kind of questions that we try to ask and answer. So this approach is has two broad phases. We have guided breadth first search, followed by assembling reactions into pathways, ultimately to identify in a static network what kind of interactions are there or exchanges are there which contribute to, say, is the organism able to grow and so on. Can you predict that? And we've implemented this in Python. What are the key features? We require only the topology of the reaction network, not even the stoichiometry or the atom mapping or the mechanistic pathways and so on. It is quite simple and scalable to large metabolic networks. So graph theoreticians usually worry a lot about cycles in the network, so wherein this can really confound a lot of graph algorithms, whereas we have an efficient method to take care of cycles and branched pathways. And it also examines alternate routes of conversion in that are possible within a cell under a given set of conditions and nutrients. So this is the first approach. So now we have a methodology to identify what kind of interactions could potentially take place. But then the next question is, how do we quantify these interactions? So how well does, say, this organism support the second organism and so on? So are we able to quantify this in some fashion? So we took a very simple approach to quantifying this interaction. We said we will compute the fraction of reactions that are currently stuck or blocked in a particular organism that get enabled in the presence of a second organism. So we compute it as a fraction. You don't need to worry about the formula, even though it's simple. But basically realize that this is like an asymmetric index, wherein A enables about 1% of the metabolism in B that was not possible for B to do on its own, whereas B enables 30% of A's metabolism that A was not able to do on its own. Just the presence of certain metabolites from B enables 30% of metabolism in A to happen in this particular system. So if all reactions remain stuck, there is MSI 0. So this metabolic support index MSI essentially tries to quantify the extent of benefit you have in terms of metabolism, in terms of what amount of metabolism is enabled in a community. And how is this useful? We found that the organisms with the higher MSI growing at a much faster rate in communities. So this is something that we inferred from experimental studies in the past, and also we also carried out the one time we did some experiments on our own to basically confirm some of our predictions. Essentially, our graph models help us address many questions. So what are the metabolic capabilities of an individual organism or community? What kind of metabolites are exchanged between different organisms? How do the organisms depend on each other? And what are the higher order dependencies between organisms in a community? What could be the unique contributors and so on? So these are some of the questions we've been able to ask and answer using our graph theoretic models of metabolic networks. With that, let me switch gears and go into constraint-based models. So a simple constraint-based model is what we built recently for designing functional-specific microbiomes from a large community, minimal microbiomes from a large community. And Indu and Ashwati led this very nice piece of work. So the idea is this. So we have a bunch of microbes that are present in any environment, and they all produce certain metabolites. But it turns out that there is a history to these microbiomes and so on. So how the organisms got added, removed over a period of time. So do all the organisms actually need to be present? Or suppose you want to build a sort of probiotic, you want to make a small intervention wherein you want to just give a few microbes as a supplement and so on. Can you actually narrow it down to like a small number of metabolites? So what we did was we basically start off with, we use a combination of various optimization approaches. We used a sequential deletion approach and also a mixed integer linear programming approach to essentially start deleting microbes that don't significantly affect the growth rate. And using this, we prune the set of microbes that can produce a given function. And we were able to identify several different microbes, microbial communities, subsets from large microbiomes that are able to produce this. So we use something called the joint FBA, which is a simple algorithm that's previously published, to maximize the community growth rate and then also maximize the production of several short chain fatty acids, and then remove organisms iteratively to ultimately come up with multiple multiple microbiomes with a similar outcome in terms of the productivity of short chain fatty acids, which are very important for things like gut health. And this is like a very interesting study we did. So we looked at large gut microbial communities and were able to whittle it down to much smaller sets, which was having very similar growth rates and like a butyrate production rate and so on. So very interestingly, our minimal microbiome does not necessarily identify all the keystone species, because there may be other, so the whole approach is it can give you multiple solutions, and this can help you identify other microbiomes that are capable of similar function and so on. So it gives you a lot of insights into potentially how these microbiomes may have evolved and what kind of species are more important and things like that. So there's a lot of fascinating things to study about microbial community structure here. I then move on to one of our most exciting algorithms where we've built what is known as a pan-genus metabolic model to counter uncertainties in microbial community composition. So if you see, there are lots of data sets available of microbial metagenomics data, but typically this is 16S data rather than shotgun metagenomic sequences, right? And if you look at only the 16S sequences, the resolution is typically only to a taxa level, but not even to the I mean the taxa is typically only to the family or genus level, but rarely to the species level. So various taxa are just mapped to the genera, but not the exact species. So with this incomplete knowledge, can you still build models? Because as a modeler, I wouldn't like to complain about the data. I would like to see what is the best that we can pull off with any given data set. So how do we build metabolic models using this sort of incomplete, uncertain data? This is the question. And there are lots of very nice constraint-based models that people have built in the past. That's like SteadyCom, OpCom, MyCom, and Joint FBA, multiple things. But they can all only work on microbial species or strains, specific strains, where you know all the reactions that are present. If you just say this is bifidobacterium and you do not know whether it's bifidobacterium, bifidum, or adolescentus and so on, how do you still can you still make predictions on what it can do, what it cannot do? And it turns out that because of the ease of sequencing and the low costs, you have a ton of 16S data compared to shotgun metagenomic sequencing data. If you knew the species, well and good, you can build more accurate models. But in the absence of that, if you're stuck with just 16S data, what is the best that you can do? This is what we wanted to explore. So we say that there is no existing tool that has effectively addressed the complexities in learning microbiome composition from 16S sequencing data and using it to build meaningful models. So the question we had is, can we build genus-specific metabolic models, yet mitigate the uncertainty in inferring metabolic capabilities? So this was a very fascinating question Indu and I had. And what we ended up doing was we built what we call PGMMs. So those in the field would have heard of GSMMs, the genome scale metabolic models. But we now go ahead and construct pan-genus metabolic models, which are essentially some sort of species-aware models which can understand the metabolic capabilities of a desired genus. So it can tell you what is bifidobacterium expected to do if it is present. Right? Not bifidobacterium bifidum, but you know there is bifidobacterium. You don't know if it is bifidum or adolescentus or whatever. What can it still do? Right? And it can also, but suppose you have some result data or you have some high-quality data for like certain species for some reason, you can also integrate that into our model. So we employed three main steps. First, we built a combined model from all the reactions, right? So it's like a full bag of all reactions. And then we have a biomass objective that captures all the species in the genus. And then we add a parameter to accommodate the variation in species composition. So you know that there is X of this, Y of this, Z of the other species. So can you then plug that into the model to alter the model suitably? So what we have is the biomass reaction for the PGM is like a linear combination of the other biomasses. And we also have a reaction species, we have a species probability vector that we incorporate as a reaction species matrix to essentially tell the model how much of each species is actually expected to be present. Okay. You can start with a uniform composition, but what is interesting is, can you still use this PGMM to explore the metabolic activities of a genus as a whole? And how does the metabolic potential vary with species proportions? We find that certain reactions do not vary much. Right? So certain other reactions would definitely vary. But let's say if you are able to predict certain reactions, and there are some important reactions like short chain fatty acids within a narrow region, then that's very interesting, which is what I'm going to talk to you in a moment. And can you employ this kind of taxonomic data to study and predict the functional abilities of personalized microbial communities? Right? Let's say you have 600 sample data from different patients and so on. Can you have a personalized map of their gut metabolic profile and things like that? So I'll cut through some of this. Let me leave out some of the technicalities. But the idea is basically this. So you have this metabolic network with different species compositions. But when you plug these in, you will get some metabolites that have low variance, some metabolites that are high variance, and some that are very, very tight. Right? There is almost no variance. It's just like very, very tight. Yeah, please do stop me at any point and ask questions, and I'll be very happy to look at it. I'm looking at my slide deck, and I'm also looking at the chat box, so I should be able to answer questions as they come in. Okay, so yeah. So these are some metabolic landscapes, and you see that for certain in bacteroids, you find that acetate and in two different diets are very tightly regulated. So here is the diet, EU diet and Mediterranean diet, and here is bacteroids, and this is acetate and this is L-cysteine. And you see that some others have low variation and some are bimodal, but they essentially, like Pravatala, the acetate, depending upon the species, it could like really vary over a complete spectrum. But what is interesting is, no matter what bacteria you have, a certain bacteroides, you will acetate is going to be very narrow, even if cysteine is going to be more in a much larger band of variation. And we found that when we built these distance trees using metabolite similarity versus reaction similarity versus flux bandwidth similarity, we found very different interesting results, and this is, I think, quite insightful for us to try and understand what's happening. Okay, and we also have hybrid models that can use a combination of these high quality genome scale metabolic models and lower resolution PCMMs and so on. And we basically evaluate these against our conventional GSMM based communities. That's the best we can do. And we found that our PGMMs can predict the functionality of communities much better than the PAN models. Nearly a 10% reduction in prediction error in various kinds of communities, even much larger communities with size 50 and so on. But there are, of course, limitations to this kind of approach. The first thing is how good are your source models? We use tools like Agora. There is also a new system called Apollo, which has like lots more models. These are all like automatically created models, so not like very high quality, but they have very consistent annotation, so it's easy to marry these models and have exchanges happening and things like this. So that's quite helpful. But I believe these limitations can be conquered over a period of time as more and more data accumulate. And to summarize this part of the work, there are not enough methods that can exploit amplicon data, and existing methods simply lumps models to literally like a bag of enzymes kind of thing. Whereas we have very adaptable PGMMs that can be very hybrid essentially in terms of the resolution of some organisms being captured at a genome at a full species level versus other at a genus level, and we can use these to build much better community models despite the uncertainty in taxonomic information. So with that, I would like to conclude my part on the algorithms and delve into some of the exciting applications we've had. I presume I have another close to 10 minutes, right? So I will go through a few applications and then open the floor for questions. So the first of these is this very exciting paper that we had again with Aarti and our collaborator Niranjan from A-Star Singapore. And what this cartoon at the center really captures it nicely. This was developed by our colleagues in Singapore. And What this shows is this likens antibiotic treatment to a forest fire. And what you have is once you have antibiotics, your gut is just nuked, like with the forest fire. And based on that, what happens is that, and after a while, as time passes, the gut slowly recovers as the ecosystem regain, right? So you have the first shoots and then back to a full-fledged forest. Hopefully, not one more antibiotic administration in the middle. So how does this gut microbiome recover? It turns out that certain organisms are able to accelerate the recovery of certain individuals. So there are individuals who have, if they have certain microorganisms in their gut, they're able to recover much faster. So they identified 21 species of bacteria called recovery-associated bacteria. And it turns out that they work very well together. And why do they work very well together? It turns out that the metabolic exchanges and interactions may play a role. So we saw that the recovery associated bacteria, they had much greater metabolic support from each other than so they worked as a nice team to get the community health back. And we also found this very specific and important interaction between Bacteroides theta iota omicron and Bifidobacterium adolescentis, and we found that this is one of the highest values in our of interaction. And our collaborators evaluated administration of these two in mouse models and saw that whenever you administer these two together, you get a much faster recovery of the microbial diversity in the gut in the mice. So essentially, this emphasizes the how you can use these kinds of models to identify key players and use it to potentially ultimately design probiotics and so on. This is another study where we looked at lots of interactions in the gut. So I will just skip through some of these things. Basically, we looked at how the interactions vary across different media and what kind of exchanges can happen and so on. In the interest of time, I think I'll just skip this, but focus on one very exciting study that we did. So this is, I like to otherwise title my talk, Microbiome Systems Biology from Deep Sea to Outer Space. So this is the outer space part, and then I do have the deep sea coming up in a couple of slides. So here we try to understand key metabolic interactions in the International Space Station microbiome. Wherever humans are, microbes are there. So, and the space station is no exception to that. And what kind of microbes are present there, and how can we understand their interactions? So we started focusing on this very specific pathogen called Klebsiella pneumoniae. You might be familiar, or you can guess that it is involved in pneumonia. And what we did was to look at viable organisms. This is an important aspect because typical metagenomic studies just pick all DNA that's lying around, floating around. But this looks at a PMA treatment to only detect those that are actually actively growing and surviving. And we looked at those locations in the International Space Station, about like 8 locations were sampled. And we looked at those locations with a significant presence of Klebsiella pneumoniae and looked at what are all the organisms that are co-present with it. And then built models to predict what kind of interactions may have. We did a network analysis based on MetQuest and MSI, which I already explained. And we also did some constraint-based analysis and so on. So we were able to identify, based on our pairwise and community interaction studies, we were able to identify that some Pantoia species are highly dependent on Klebsiella for survival. And we also identified many parasitic interactions or amencillistic interactions and so on. So this shows Klebsiella pneumoniae having a negative effect on Penicillium rubens, Aspergillus niger, Penicillium nalgiovens, and so on. And what we captured here is that we also did some experiments. Our collaborators did experiments. These are electron micrographs of an Aspergillus, closely related to Aspergillus, with Klebsiella pneumoniae in normal gravity and microgravity. And it turns out that when you grow the two together, in microgravity, in the sort of simulated space environments, the effect on the fungus is massive. It's a very strong negative interaction between the microbe and between the Klebsiella and the fungus. So how does this? So these are basically very interesting because you have a hypothesis that is computationally generated, and then you go and test out in a wet lab. and you see that even if you can't explain everything that's happening, there's still a very interesting interaction that you have now uncovered. So this is always a challenge, though, that we have lots of hypotheses for experimental testing, but ultimately they can provide a lot of insights into how clean room microbiomes function, and how do you potentially disinfect or disrupt them, and so on. That a more recent study of ours is about understanding multi-drug resistance Enterobacter buchanensis and how it persists, and what is its succession about the International Space Station. So here we did a bunch of bioinformatics studies to do a pangenome analysis of different isolates, both terrestrial Earth isolates and also those from the International Space Station, and looked at what are their functional differences, and differences in virulence, difference in resistance causing genes, and so on. I do see a question on AMR in the chat. So it turns out that the space microbes, the space station microbes, are mutating much faster, probably because of the extreme conditions of heat, radiation, CO2, and so on. And they are acquiring various mutations much faster than what we find on the terrestrial strings. And we again did a bunch of analysis in terms of what are the dependencies between Enterobacter buchanensis and other organisms. So we find several negative interactions, positive interactions, and things like that. And we also identified several new important new genes and markers for pathogenic strains about the ISS. Right? We identified as many as 14 MDR genes, gene classes in the ISS strains, and several interactions between Enterobacter and other organisms, including Klebsiella pneumoniae. Right? So these pathogens try to somehow feed off of each other and cause more trouble ultimately. And this is one of the last studies I want to touch upon. I think I'm almost at the end of my talk. So this was done by Dinesh and Karthik Anantaraman, who's like my collaborator in Wisconsin-Madison. He's the speaker of the talk I was just mentioning in the beginning of my webinar. And Shreyansh from IIT Madras was also involved in this. And here we looked at this biomass basin, where you have these are hydrothermal vents, are basically underwater volcanoes, where you have this hot 400 degrees Celsius hydrothermal fluid that meets very cold ocean water. And what you get is like a very interesting gradient, where you have all kinds of cool extremophiles with exotic metabolisms and things like that. So how do these microbes interact with each other, and what kind of dependencies do they have, and so on. This is very interesting because you can't even culture many of these things. So modeling is perhaps the only way we have to even make certain guesses about how they function and things like that. And we again looked at metabolic dependencies between the organisms, and how the various kinds of like say microbiomes differ from each other in terms of we computed something known as a metabolic resource overlap, based on their metabolic networks and so on. I'll not get into all the details, but those who are interested can go and read more about in this very interesting paper that Dinesh, Shreyansh, and others worked on. So with that, I should underline the limitations of this approach. First thing is that the graph models are pretty static. They don't really capture some of the dynamics, but they are very useful in terms of identifying what could be the possible exchanges, no matter what the environment is. And then we can use constraint-based modeling to see, as the environment changes, how these exchanges or interactions may change and so on. So you can look at graph theory and constraint-based modeling as very complementary approaches. And we find that some predictions agree with previous experiments, but the pace at which we generate experimental suggestions is far higher, right? And the pace at which we generate this hypotheses for experiments is far higher than we can do experiments. And even in the MetQuest algorithm, the quality of the input network is very important, and we don't yet have a way to weight or rank paths. That could be an interesting upgrade to the approach. And last but quite importantly, we view cellular interaction only through a metabolic lens, and potentially a lot more is happening. But that said, I have time and again emphasized in my talk that the metabolic interactions are very crucial to understanding how microbes survive and interact with one another. So this is probably a good way to look at it, and maybe not too limiting after all. Some very interesting questions. What are the key differences between pairs of microbiomes? Could be healthy or disease, or rural or urban, healthy or breached florals, and so on. So we're trying to do some differential analysis of microbes and microbial association networks, trying to understand microbial trajectories. How do these communities assemble? What are their keystone species? How stable are they? What kind of trajectories that can they take? Can we manipulate them using prebiotics, probiotics? All of these are exciting questions that we are looking at. And there are many challenges in terms of understanding the dark matter. There's a lot of underground metabolism that we still don't know. So how do we figure out what kind of reactions are happening? And we still seem to know very little about the viruses that exist there, or the fungi that exist there. We are all often, we should stop saying, many of my colleagues in the field say that we shouldn't be saying microbiome, we should be saying bacteriome, because we really aren't looking at the other microbes or even the archaea many of the times. And how do you experimentally validate these models, including the exchanges and so on? It's very difficult to do C13 labeled flux analysis and things like that. And the other challenge is how microbiomes have a lot of plasticity, in the sense, they can fluctuate a lot without the function changing. And then there's also geographic variations, individual to individual variations. So you have so much variability between healthy people themselves. So how do you ultimately predict who has disease and who is healthy and so on? And of course, lots of data challenges and things like that, or even studying host microbial interactions and so on. And there's always a challenge. This is broad to the whole field of computation, modeling, and science itself, in terms of reproducibility of experiments and reproducibility of the models and so on. So this is something that again we are interested in, and we are trying to address this through various frameworks and things like that. And we also need to have better resources for modeling and better algorithms. So this will greatly impact the field. So this is just my 2 cents on this. And I'd really like to advocate for building a much stronger community in India, especially. And I think we have the nucleus for that. We have run a few symposia where we have lots of exciting participants, a lot from India, and looks like there's a lot of promise for this field. And so this is like a quick summary. I think I mentioned everything. So I'll just say the last sentence, that metabolic interactions drive community structure from deep sea to outer space. So we have a lot of generic frameworks to understand microbial interactions, identify exchanges, and have insights into how microbes interact with one another in various microbiomes. And I'd like to acknowledge a lot of people without whom this work would not have been possible. So lots of students, collaborators, postdocs, faculty from other universities who've been very instrumental in developing our research program, and also all our funders, without which this would not have been possible. With that, I'd like to pause. Thank you all for your attention, and I'm very happy to take questions.
Thank you, Professor Karthik, for that superb overview of the exciting research happening in your lab. So over to questions.
So there's already a couple of questions. I'll start with that, and maybe others can unmute themselves or turn their video on and ask me questions. Yeah. So there was a question: can you use GSMM to study AMR genes within species? So AMR genes are typically some sort of regulatory sequences and so on, and although some of them may have some metabolic function, so I don't see how you can specifically use genome scale metabolic models to study AMR genes. But there are other approaches to track resistance and so on, and we've also, I've done some work during my PhD, and I have a new intern who's trying to build on some of these things, trying to essentially understand how resistance develops in microbes by studying their protein interaction networks and so on. And there's one more question, which is on a clarification that in one of the applications, under normal conditions, Aspergillus and pneumonia have less effect, and under microgravity, it has a huge impact. Yeah. So, yeah, we are really not sure about what are the factors, but it just looks like that, the strains that you have in the microgravity environment and so on, they have picked up mutations and so on, which are potentially having a stronger impact. So, yeah, we don't know enough about this, but I think what we think is that there is a very strong, there are other stresses in that environment that is further contributing to it. It's potentially even outside what we modeled, but it just seems like the rate at which these organisms grow is heavily dictated, is more strongly dictated in these extreme conditions that you find in microgravity and so on. Yeah, happy to take more questions. So keep them coming in the chat, or turn on your video and ask.
More questions, please.
It's a very good question from Sashikala, that do we consider pH, temperature and environmental factors for modeling microbial communities? So essentially what we consider are the microbial reaction networks, and they change with pH and temperature. So if you have a way to, so that is the lever that you have to move, right? So if you change the pH or the temperature, the network is expected to change. Or ideally, if you had transcriptomic data corresponding to that condition, you could potentially integrate it and so on. And environmental factors, we obviously pay a lot of attention to the medium and the nutrients and so on. But other environmental factors, we could potentially include it by means of these transcriptomic data sets and so on. We don't actually yet model these things. It is definitely doable by integrating metatranscriptomic data or things like that. So those would be very interesting lines of research.
A lot of open questions still in the field.
Yep. Lots of open questions, and I think that's why we need lots of youngsters to come and work in these spaces, right? So, right, including what I was just talking about, the dark matter, right? What we are, everything that we are talking about is by analyzing maybe 30-40% of the data. We don't know how much we don't know. So that's the really exciting part of it. And we also aren't giving enough attention to fungi and viruses. And they have impact everywhere, from ocean to soil to gut and so on. So I think we really need to start reanalyzing these data sets, and there's a lot of work to be done.
So, like, I had one question. How much of an effect would these compositional shifts in the microbial community? So, I mean, like, they are supposed to hold the key to gut health and gut disease. So, in most of the environments, these compositional shifts would alter the entire community. And so, do you think that's a very intrinsic aspect of modeling that should be taken into account going forward? Or do you think still graph models?
Definitely, right? So which is why we pay a lot of attention to the environment for these microbes, right? So how does this influence but the biggest interesting part is that there is just so much of flexibility in these microbiomes, right? So anytime we do a PCA kind of plot of healthy and diseased microbiomes, you have them spanning the entire space. So it's not that healthy is like very structured and then disease is much larger or anything like that, or vice versa. What you see is everything just intermingles. It's so difficult to deconvolve them and remove. But then off late we've been seeing some interesting. We've been developing some custom features. We like to do a bit of feature engineering on these machine learning data sets and things like that. Wherein we are looking at the ratios of different species and things like that, it does seem to have some interesting ways to deconvolve these different data sets and so on. So I think to answer your question, the composition of these microbiomes is definitely important, highly influenced by the environment. And I suspect that we need to have newer methods to see. Definitely healthy people are healthy and diseased people have disease. And there is a lot of influence of the microbiome on that. So I think we need to go over it with a fine-tooth comb to pick what are the key differences that are leading to this, and at least start with some correlational studies, features, and then go and investigate causation. But this is like super exciting. Yeah. Okay. Just a moment. Yeah. Yeah, I see some very nice questions. So, Yeah, so short chain fatty acids are widely used. I think that we can. There's also a lot of additional amino acids and things like that. But if you see this acetate butyrate and that balance is like very heavily investigated, and especially when you're looking at gut health and so on. And in terms of bioinformatics approaches, we use a lot of these tools, right? There is like Metaflan, the chain pipeline, and so on. All these are very essential in the initial stages of metagenomic data analysis. Following that, we then use standard metabolic modeling. Ultimately, get genome sequence, put it into K-base, get a model, tune it, predict the interactions. But the initial part of actually finding out, filtering out, sifting through what's there in your data set and what is the organism that I'm supposed to be looking at. Are these MAGs and so on, see metagenome assembled genomes and things like that. These are things that we need to look at. And how does min microbiome algorithm optimize microbial communities for SCFA production? So I sort of ran through the algorithm, but the idea is we say we take the original microbiome, that is given microbiome, and compute what is its SCFA production, and we use it as a constraint to find out by removing other species what kind of removals are going to not affect this, and we do it computationally efficiently to figure out that a much smaller set of microbes is also able to ultimately produce the same approach. And in terms of that's a very good question from so thanks Mohita for the question, and Abhishek has another very good question, which is like what are the challenges you are facing in data sets for ML? Lots of challenges, right? So first of all, the data sets are small, and then the challenge is a lot of traditional microbiome modeling typically involves looking at abundances and species and things like that. But that is not where the function is. Right? So the function is in the SCFAs or in the metabolites that are being produced and so on. So we are literally looking at the wrong space and trying to cluster and try to make sense and so on, which is why I suspect it's not working. But not that we have really found an answer to this. I can tell you what's not working, but I don't think I can still tell you enough of what is working. So that is one challenge. And then of course, the noise in the data, the batch effects, all of these things are other problems. So there's a whole litany of data cleaning problems that one has to solve when you start working with microbiome data sets for machine learning. And Yamini has a question. So as microbial systems are complex, which metabolic modeling is conveniently preferred to influence MDR for predicting resistance and metabolic fluxes, and how to distinguish which algorithm is best? So I mean, I don't know if I understand the question fully in terms of how do you influence multi-drug resistance, because we try to predict some of these. There are other ways to predict the maybe sequences and what kind of mutations may develop and how they may contribute to drug resistance. But I'm not exactly sure how we would predict them from metabolic fluxes, right? And in terms of what is the best algorithm, I think that answer is I would say is simpler, so to speak. It really depends upon the question that you're asking, right? So if you're looking at some static thing, you can use a network model. If you really want to understand what kind of interaction is happening, is there a parasitism or what are the relative growth rates and so on, you wouldn't need to go for a constraint-based model and so on. So I would always argue that the nature of the question will determine the modeling approach that you will have to take, and if possible, use multiple complementary modeling approaches to try and understand your system better. Okay. Thanks for the question. So I think Dr. Suma has a very nice, important question. What are the industrial applications and potential opportunities for metabolic modeling? And well, I have several ongoing projects with industries. So most of these projects are in terms of understanding systems. I don't think we yet have a project in the space of optimizing a metabolite and so on. Although there is a lot of we've had a lot of success in our lab scale experiments and pilot experiments on that. And we hope we will be able to translate some of them to the industry shortly, because we now have a big center of excellence coming up in the space with Herbalife. And I think, this is definitely that. But ultimately, metabolic modeling is very crucial, and it can give you lots of insights into how to manipulate microbial systems for overproducing metabolites or microbial co-cultures potentially, and so on. So I think there's a lot of promise there. It's really waiting to be explored and delivered. There's one more question on, so if the data set is small, does it capture the very essence of the real-time environment perfectly? Well, there are definitely challenges. In fact, yesterday I was discussing with a collaborator about air sampling, right? So you see that there is a lot of microbes that are airborne and so on, and maybe in certain areas, like say near sewage treatment plants and so on. So how do you sample it well, right? And so this is definitely a challenge. So how do we do the sampling that is reliable and sufficient? But at least if you look at the International Space Station, we have very good data collection that is well spaced out, and so across 3 different time points in 8 different locations and so on. So we have like a fair understanding of what the microbial load is. And there's also a very exciting program called MetaSub, where they started over with this New York subways back in 2015-16, I guess. And they've been able to identify signatures there, and they now still continue to do a global city sampling day on the 21st of June every year on the summer solstice, and trying to figure out what the microbial load is in different cities, both in the Western Hemisphere, both in the Northern Hemisphere and the Southern Hemisphere. Right? So, so the thing is, I think we always would like more data, and because this will shed, and there are lots of studies that are also showing that there is a lot of fluctuation in microbiomes. So I think the more data we have, the much better placed we are to answer many of these questions. And in another point, it's also important that if you have, because just because you sample these places, you cannot assume that these microbes are very rare and so on. We also need to understand the presence of these microbes across various environments and things like that. So this again is, I don't think there's an easy way to do that yet. And this one question from Kanishka Mohan, who says, what is the biggest challenges in combining omics data with metabolic models for drug discovery? I think that is definitely doable. The challenges are mainly in terms of availability of the right kind of omic data and also good algorithms. So I've seen very good cancer models wherein you have metabolic models and transcriptomic data, so you can really integrate them and understand in a tissue-specific fashion what is the change happening in a cancer cell versus a healthy cell. So if you can have a better handle on the etiology of that cancer, you may be able to better target that disease and so on, right? So that's definitely doable. I think the challenges are in finding the right algorithm to work with the right data. Like I said, right, like for example, Panera, if you have 16S data, a ton of it, and you don't have a way to meaningfully use them, you need to really figure out what is the best way to use it. You need to build new algorithms to leverage existing data. Because on the one hand, you can keep complaining that we need better data, we need newer equipment, we need more money, and so on. On the other hand, what is the best use that you can make of existing data to make meaningful predictions and so on? Okay. Yamini has another question. So suppose we have to predict multiple combinations of various antibiotics in case of drug resistance. What methodology or algorithm you should opt for? This again is like a very open-ended question, right? So you can use. I like to ask these kinds of questions in my exams because you can really think of various ways to do this. But one can think of so one approach is sort of looking at the sphere of influence of each antibiotic and see if it is targeting completely, which antibiotic is targeting completely different set of pathways and things like that. There's a very good drug combinations paper by Barabashi. Barabashi is a famous name in this networks field, and they have a very good paper. And there is also one more that appeared in, I think, Cell Discovery or so in 2021. If you want, you write to me and I can share some couple of these interesting references. I've also discussed some of these in the last chapter of my book. Okay, so then there's a question by Dr. Devika, which is, can Panera predict the metabolites secreted by a microbial consortium derived from 16S rRNA data? And potentially, yes. Validating it is going to be a challenge, but basically, in fact, what would be interesting is if you can, this is a very good question. So what we could do is we could build these community models and look at what are the exchanges happening between the PGMM, the pangenus models or different genera, and look at those that have a narrow bandwidth, meaning they are very, very likely to happen because it doesn't change much with species composition. So those may be a very interesting place to start out. So yeah, but that's like a very interesting thought, and we could potentially, use it to. I don't know. It's probably more interesting to think about. Can you identify metabolic difference between healthy and diseased? That remains to be seen. So in fact, this is something that I was coming to mention in the context of ML. So can we use more meaningful features like metabolic features and fluxes and things like that to make predictions? Because just abundances of microbes is not going to be sufficient for the most part.
I think let's, more questions. I think you can email Professor Karthik, and he's would be very happy to respond to all your questions. I think it's getting a bit late in the day, so thanks for sharing more details on your research. Now I call Dr. Suma to propose a vote of thanks.
Thank you, Dr. Ashok. So good evening, everyone. On behalf of Shastra University, Division of Bioinformatics Center and Compic, I extend my sincere gratitude to Professor Karthik Raman for delivering such an insightful session on metabolic modeling. So the session was truly enriching, and I deeply appreciate the time and effort you dedicated to sharing your knowledge with us, sir. We sincerely thank sir for graciously accepting our invitation and for his valuable insights and encouragement. Next, I extend my sincere gratitude to Professor K.S. Rajan, Dean SCBT, for his support and encouragement in organizing this webinar. I would like to thank the members of the Combic students team for the arrangements and the members of the BIC and Bioinformatics division for the support. I thank all the participants from academia, industry for their enthusiastic engagement and making this session a meaningful and interactive learning experience. Thank you, Dr. Ashok sir, for hosting the session. So once again, I thank you, Professor Karthik Raman, for being part of the webinar series. We look forward for much such interactions in the future. Wishing everyone a great evening ahead. Thank you, sir.
Thanks for organizing this series, and I look forward to the upcoming webinars as well. Thank you.
I hope you can join us for the upcoming webinars. So I'd just like to remind all the participants that the next one is scheduled on March 15, and the time is from 7 to 8. We have a speaker from the US, from Sanofi US, Dr. Shamir Kader, and he'll talk to us about AI applications in healthcare. So keep watching this space and join us then. I'd also like to thank Professor Karthik for leading the systems biology community in India. And thank you so much for supporting our department and our school. Thank you.
Thank you.
Thank you, Professor. Thank you very much.
Thank you.